To our friends joining us on the recording. We are covering week number 2 lab that relates to 3 topics: regional terms, microscopy, and cytology. When we talk about regional terms, what I want you to make sure that you can do with regional terms is I want you to make sure that you can identify these regional terms on my picture. You'll notice that after or actually it's before the pictures in your lab package, you have all these words, right? All these written descriptions of where I find each of these body regions. I don't care as much that you know how to write out the definition of where the calcaneal region is found. What I do care is when you have this picture that shows you the front side or the back side of the body, that you can identify where that region is found. Because if you can identify it in a picture, if you can feel it on yourself, you'd be able to describe it in the words that we would use on the quiz. So with that being said, let's talk about some of the big regions that we see in the body. The first way that we can talk about regional terms for the body relates back to all the bones that we learned last week. So for example, let's start here with this region that's right over the middle of the chest. Do we remember what bone we find right here in the middle of the chest? What bone lives here? Yeah, several of us chiming in. This is where the sternum bone lives. So if I know that the sternum lives in the middle of the chest, it shouldn't surprise me that this is called the sternal region, where the sternum is found. Or if I know that the bone that lives here in the thigh, what's that big bone here in the thigh called? Your biggest bone in your whole body. Yeah, my big bone here in the thigh is the femur. So this is the femoral region. When you're looking at the list of all of the regional terms that you're labeling on the picture and that you're needing to know where they're found, many of them are going to look like the names of bones that we learned for last week. Use that as your guide to help you figure out where those regions are. Now, there are a couple regions that I want to make sure to review with you because those are important bones that sometimes we get mixed up. Let's talk about our 2 regions that are here in the forearm. When we're talking about the regions here in the forearm, they're named based on the 2 bones that we have here. So here in the forearm, we have the radius and the ulna. The regional terms for these areas are going to be the radial region and the ulnar region. But for me to figure out which is which, I'm going to have to remember which side has the radius and which side has the ulna. So let's start here. This region over here is the medial region, the one that's closer to the body. But the way that Dr. Aulis is going to figure it out, not is by calling it medial. I'm going to figure it out by saying, oh, this is the pinky side. When I talk about the pinky side of the forearm, which bone lives over here on the pinky side of the forearm? Is that the radius or the ulna on the pinky side? Yeah, a couple of us are remembering. The pinky side has the ulna. The way that I remember that is we've got UP, up. So the ulna and the pinky are on the same side. The radius and the thumb are on the same side. So when I'm talking about the region on top of the ulna, that'd be the ulnar region. When I'm talking about the region on top of the radius, that's the radial region. The same thing applies when we go down here into the leg, where we have a medial and a lateral region. Remember that when we're down here in the leg, the bone that's on the outside of your leg is called the fibula, the little one, like a little fib. The fibula here on the outside. The tibia is the big T-bone that's here in the middle. So the tibial region is medial on top of the tibia. The fibular region is lateral on top of the fibula. So one way that we can name the regions in the body is based on the bones that are found in those regions. Another way we can name the regions of the body, though, is based on those cavities that we talked about. So remember last week that we talked about there was a name for the whole chest cavity, for everything above the diaphragm. Do we happen to remember what we had the name for the whole chest cavity, the big one? Exactly. Yep. Several of us remembering that the whole chest cavity, this area up here is called the thoracic region. So this is called the thoracic cavity, excuse me. It's also called the thoracic region then. In the same way that this area right here was the top part of the abdominopelvic cavity. So this is the abdominal cavity that we see here. And down below, the region around the hip bones, that's the pelvic cavity. So when we're looking at the names of these regions, it's possible that we're looking at a cavity term, like we learned last week. Or it's possible that we're looking at a bone term, like we learned last week. There are a few others that won't quite line up with stuff we've talked about before. So a couple of examples of that. We're looking here at the armpit region. So the armpit region is called the axillary region. If you've ever heard about taking an axillary temperature reading, that's where you're doing that's sticking that thermometer in the armpit to take that reading. Another regional term that'll be new to us, the ear region is called the auricular region. It gets its name from something we'll talk about in the very last week of lab called the auricle. So the auricular region is named after the auricle, which is the outside part of your ear that you can see. So again, I want to emphasize, as you are working on learning regional terms, it's not worth spending a lot of time talking about or memorizing these word descriptions of where the regions are found. Make sure that we can find those regions on ourself or on a picture of a person. If you can find those regions on yourself or in that picture, you're going to be able to match up these terms as well. We're going to switch gears out of big picture regional terms, and we're going to start talking about some smaller things, some things that are going to be really important for us, especially next week as we start working together in lab. So the first thing that we're going to talk about that we're going to be doing next week is microscopy. Microscopy is a fancy word for saying we get to play with a microscope. It's going to help us see things that are smaller than we'd be able to see with our naked eye. We need something to make them bigger. Microscopes help with that. Cytology is one of those small things that we need a microscope for. When I use the word cytology, I'm talking about individual cells. So we're going to look at the organelles that are in cells, just like you read about at the beginning of the lesson number 2 packet for lecture. We're going to talk a little bit more about those organelles and identify them on our models in this lab portion here. So when we talk about microscopes, there are many important parts of the microscope that all do different things. I'm going to walk through the microscope with you, help you to label things, and also kind of describe what each of these things is doing to help you later when you're matching up the names of parts with what they do. So let's talk through the way that we would use microscopes in a typical lab class. The first thing that we would do when it's time for us to do microscopes is we're going to get our microscopes out of the cabinet. There are 2 parts of the microscope that you hold onto when you're transporting that microscope out of the cabinet. So the first part is this bottom part right here. We've got an arrow labeling it right here. This part of my microscope is known as the base. This is the base of the microscope. The base is just like its name said, it's the bottom. It's the base of the microscope. Sticking up from the base is this part here. And this part here with our arrow is called the arm. So the arm of the microscope is what sticks up. It's where we're going to see our ocular lenses attached to. When you are carrying a microscope back to your lab bench, you're always going to use 2 hands to carry it. One hand underneath the base, one hand behind that arm, holding onto that arm. 2 hands to transport the microscope, microscopes being the most expensive thing we will use all semester in lab. So we get the microscope to our lab bench, we're ready to start using it. The part of the microscope that you look through, I mentioned it briefly before, is this thing here. This is called the ocular lens. So you look through the ocular lens to see things that are on your slides. By itself, the ocular lens makes things 10 times bigger. So you could actually pull off this lens and hold it up and peek through it. It would make things 10 times bigger by itself. That's great, but it's not good enough to help us see things like individual cells. We need more magnification than just 10 times. And that's the job of these things that we see down here. So these things that we see here are our objective lenses, but they each have their own name and they each magnify things differently. So let's start with the little one. Sometimes in class together, I'll call this one red. This is the red objective lens. So the red objective lens, technically its name is the scanning objective. The scanning objective by itself makes things 4 times bigger. So it's not much bigger, it's not even as much as the ocular, but as we'll talk about shortly, when you look at things in a microscope, it's a compound microscope. So the magnification of the ocular works together with that of the objective to make it so that you make things bigger. So here's my scanning objective, my red one, that makes things 4 times bigger. Next door, we've got our yellow one. This is called the low power objective. The low power objective makes things 10 times bigger by itself. So it magnifies things the same amount as those ocular lenses do. Our final objective, the one that works the hardest, is called the high power objective, the blue objective. The high power objective by itself makes things 40 times bigger. So when we're looking at the teeny tiny cells that make up your skin, for example, we probably are going to have to take those all the way up to the blue objective to see them clearly. When we're looking at a tissue like bone, for example, bone tissue, we can probably just do the low power objective. There's not going to be a lot of slides that we use the scanning objective for by itself. Most of them have to go up higher. But the scanning objective is where we always start when we're looking at specimens. All of the objectives are attached to a thing we can see right here with these little lines on it, but you can also see it labeled on this picture over here. This is something called the revolving nose piece. Like its name suggests, the revolving nose piece moves in a circle. So it allows you to start with your red objective pointing down and then turn it so that the yellow objective is pointing down and turn it even more so the blue objective is pointing down. So the revolving nose piece allows you to pick the objective you're planning to use. When we're looking at something, we need a place to put it. That's the job of this big black thing we see right here. This is the mechanical stage, this big black thing, and we see it over here as well, the mechanical stage. So this is what I put my slides on top of. This is what holds the slides. This mechanical stage also has the ability to move up and down. We'll talk about how we move it, but when we move the stage up and down, that makes things more or less focused, the mechanical stage. On top of the mechanical stage, there's something I can see better over here in this picture, and this is called the stage clip. So the stage clip is where I clip my slide into place. Because when I am looking at a slide, I might need to move around left and right, forward and backwards on it. Maybe when I loaded it onto my microscope, I'm focusing on the label, which doesn't have the tissue I'm looking for. I clip that slide into place in my stage clip, and then I can use these things hanging down over here, my stage adjustment knobs, to move that slide back and forth, left and right. So that stage clip is also what we see right here. It's a little bit harder to see, but this is the little lever you push on to open it up so that you can put the slide in place. So the stage clip holds that slide in place. on top of the mechanical stage, which is where the slide rests on, and we can use those stage adjustment knobs to move it forward, backward, left, and right. When I'm ready to move it up and down, I need to use the knobs that I find on the arm of the microscope. So on the arm of the microscope, there's two knobs that you're going to use. The first knob is the one that's really close to the arm here in the back. This first knob right here is called the Coarse adjustment knob. And the Coarse adjustment knob moves the stage a lot up, a lot down, the Coarse adjustment knob. The Fine adjustment knob sticks out a little bit more, and the Fine adjustment knob just moves the stage in little increments, teeny tiny, you may not even be able to see it. The Coarse adjustment knob is what I use to first find a specimen on my slide to get it mostly in focus. When it's mostly in focus, then I'll use the Fine adjustment knob to get it all the way there. I'll mention that when we look at the microscope from the front, you can also see the Coarse adjustment knob right here, and you can see that Fine adjustment knob over here as well. So we're only labeling them on the side view, but here's that Fine adjustment knob that sticks out a bunch. Here's that Coarse adjustment knob that's closer to the arm. Just a couple of other things to mention for you. If we're going to look at a slide, we're going to need a light source. So underneath your microscope, underneath the stage, you have your light source that shines light onto the slide. You can adjust how light or dark that light by itself is using the light adjustment knob. So making the light brighter or dimmer, that's what this one does. But we also have the ability to decide how much of that light or dark light we're letting into our slide. And that's the work of this thing that we see right here. So hanging down underneath the stage, we have something called the iris diaphragm. That's this lower arrow that we see here. The iris diaphragm works like the iris in your eye that expands or contracts to determine how much light is going to pass through. So the way I can make that expand or contract is using this little iris diaphragm lever. Very easy name for us, right? The iris diaphragm lever moves left and right to open and close the iris. That's going to determine how much light gets up onto the stage. There's one other thing that we can't see in our pictures, so we don't ask you to label it, but we do ask you about what it does. Up here above the iris diaphragm, there's one more thing on a microscope called the condenser. And the job of the condenser is to focus the light on your specimen. So the condenser takes however much light the iris diaphragm let through, the condenser focuses it and gets it to where it needs to be. So one last thing that, again, we won't ask you to label it, but we do have to know what it does. The condenser, which is found up here, focusing that light. I'd like to pause for a moment and give you a chance to ask any questions that you have, or feel free to shoot me an emoji. What are we thinking about microscopes? I'm gonna even send you one too. Questions and emojis. There's Dr. Ollis's. I love it. Shelby's feeling the science. Good. Hannah's processing. I like it. Very wise. So I suspect Noah is asking about this knob down here, if I'm interpreting correctly. Can I ask my friends in the chat to help me out? What did we call this knob down here? Do we remember? I talked about it kind of fast, so let's see if anyone caught it. Yeah, so here's that light adjustment knob. That's this one right here. the light adjustment knob that rolls backward and forward to control the amount of light. We were talking about, a couple of us mentioned our coarse adjustment and fine adjustment knobs. That's these ones over here, was the coarse and fine adjustment. So the coarse adjustment knob was the one close to the arm, and the fine adjustment knob was the one that sticks out a little bit more. And I know a couple of us mentioned objectives as well. So remember, the objectives are the ones that have the colored stripes on them. They're up a little bit higher on our revolving nose piece. So good review of a lot of different things that we looked at. Absolutely. Yeah, so Logan asked a good clarifying question about the difference between the coarse and fine adjustment knob. The coarse adjustment knob moves things a lot. The fine adjustment knob moves it just a little bit. And actually, here would be a good note that I want us all to write down. So let's find a place on your paper where you can write down a note, because this is, like Dr. Alla says, this is an underline, highlight, star idea, something important. Here's what I want everyone to write down to get us ready for next week with the microscopes. We're writing this down. The coarse adjustment knob can only be used with the scanning objective. The coarse adjustment knob can only be used with the scanning objective. Yeah, Madison beat me to it, right? Madison's on top of it. We can only use this coarse adjustment knob that moves the stage a whole bunch. We can only do that when this red objective, this scanning objective, is in place. Because once we start putting these other objectives in place, we run the risk of potentially breaking our slide, because we could bump that slide all the way into the objective and literally break it. So when we talk about that coarse adjustment knob, we can only use that with the red objective, with the scanning objective in place. That's actually a good place for me to mention one of the terms that we'll come back to later. The amount of space that you have between an objective lens and the stage. This amount of space, I'm going to type it in the chat. It's something called working distance. Working distance. The amount of space between the end of your objective and your slide that's on the stage. When we have different objective lenses in place, the size of our working distance changes. So my little objective, my little 4x scanning over here, when I have that pointing down, lots of space. We have a big working distance. When I put down that 10x objective, we go from something that maybe was this long to something that's this long. Now we're taking up more space. And especially when I put in that high power objective, it's almost scary how small the working distance is when we put that high power objective in place. So perhaps the biggest reason that we cannot use the coarse adjustment knob when we have yellow or blue down is because there's a very good chance we'd break our slide. The other reason, though, I'll mention that we wouldn't want to use the coarse adjustment knob is because our microscopes are something called parfocal. Parfocal. Parfocal means if you get it in focus with the red objective, when you go up to yellow, it should almost already be in focus. Maybe not quite, but almost. And if you get it in focus on yellow, you should almost, not quite perhaps, be focused on blue as soon as you rotate it over. So parfocal microscopes mean all I have to do is fine tune or change just a little bit of my focus to help me see things clearly. That is not possible with that coarse adjustment knob. That coarse adjustment knob changes things too much and wouldn't allow you to keep things in the space they need to be in. So a couple of terms we'll come back to later, but I wanted to mention them now: the working distance and the idea of parfocal. Yeah, so Hugh asked about total magnification. We are getting there. To answer your question briefly now, we're never going to go higher than 400 times in our class. If you're taking microbiology, there is actually another objective that those microscopes would have on them that we do not have in our labs, since we don't look at anything quite as small as the bacteria. But yeah, good question. All right, so I'm not going to go through and match up functions. Want to remind you that these are not matched, so don't like frantically scribble, this is incorrect, but we've talked through the functions of all the microscope parts, and you have that online guided lesson that has those parts as well. What I want to move into more is the ways that we use our microscopes and some more of those characteristics of them. So we already talked about this when we were talking about our knobs. Remember that you have a coarse adjustment knob and a fine adjustment knob, and which of those knobs you would use depends on which objective you have pointing down. So remind me, because I already had you write this down. When we're talking about the scanning objective, when we're talking about the red one, can I use the coarse adjustment knob, the fine adjustment knob, or both? What do we think? When red is pointing down, coarse, fine, or both? Yeah, being a little tricky on you, aren't I? I'm thinking, So when we have the scanning objective in place, this is the only one that we can use the coarse adjustment knob on, but we can also use the fine adjustment knob too. When the scanning objective is pointing down, both of them are fair game. We can use the one that moves the stage a lot or the one that just moves it a little bit. We can use either of them. So with the scanning objective lens, total freedom. You can use either of those knobs. But when I get to the low power objective, or especially when I get to the high power objective, which ones can I use at this point? When the yellow or the blue is pointing down? Coarse, fine, or both? If yellow and blue are pointing down. Exactly. I can only use the fine adjustment knob. Once that, to use one of those terms from before, once that working distance gets really, really tiny, you only want to use the fine adjustment knob to move things. So for low power and high power objectives, yellow and blue, only use the fine adjustment knob. For the scanning objective, red, we can use both of them. So an idea that was mentioned earlier in the chat was the idea of total magnification. So I mentioned this term briefly before, but I'll mention it again here. We work with what are called compound microscopes, and it being a compound microscope means that every time we magnify something, we actually magnify it twice. So the first thing that's making things bigger are those ocular lenses, those things that you look through. The ocular lenses on all of the microscopes in our lab make things 10 times bigger every time. We always are making things at least 10 times bigger. But we're also magnifying with those objectives as well. And remember that the objectives each have a different power. So red makes things 4 times bigger, yellow makes things 10 times bigger, blue makes things 40 times bigger. If I want to know how much bigger what I'm seeing through my objective is compared to the size of that thing on my microscope slide, I have to do a little bit of math. Now, Dr. Ollis will not ask you to do very much math in our class time together. It's not a math class. But this is one set of math that we will have to be able to do for quizzes, for the exam. It's a calculation of the total magnification, or how much bigger I'm making things compared to how they look. The good news is it's a really simple calculation. We take the power, or the number for those oculars, and we take the power from the objectives, and we multiply them together. So every single time, my oculars always make things 10 times bigger, every single time, because the oculars don't change. But the objective power will change. So when I have that red objective pointing down, remember that the red objective by itself made things 4 times bigger. Or when I have that yellow objective down, that one makes things 10 times bigger. Or when I've got that blue objective down, the blue objective by itself going to make things 40 times bigger. So if I want to know the total magnification, just how much bigger we're actually making things with each of these objectives in place, I have to do 10 times 4. It's simple math. Help me out. 10 times 4, what would my total magnification be? 10 times 4. Exactly. Yep. Your math teachers are like, I'm glad that you learned this. This is good. So when the red objective is pointing down, what you see through the oculars is 40 times bigger than what's actually on the slide. Okay, that'll help me find something like the general location on the slide, but I can't see what I'm looking for. So then I got to go to yellow. And when I'm doing the yellow objective, 10 times for the ocular and 10 times for the objective. My total magnification with the low power objective in place, 10 times 10, which is exactly, yep, Marlene chiming in, Logan chiming in, 100 times bigger. And again, I mentioned that some of our tissues that'll work for, like bone tissue, we're golden. 100 times bigger works. Most things, though, we got to use that blue objective pointing down. And now we're doing 10 times 40, which is 400 times bigger. So really small things, we're going to have to go all the way up to blue to see them. For these kinds of questions on the homework assignment and on the quiz and the exam, either you need to be able to do some quick math in your head, do 10 times 4 or 10 times 10, 10 times 40, or you can memorize the total magnification. I don't care, whatever you'd prefer, but these are a set of numbers. I don't make you learn a lot of numbers. Lost my pointer here. I don't make you learn a lot of numbers, but I will make you know these ones, the total magnification with each of these different objectives down. How do we feel about total magnification? Thumbs up, thumbs down. Any questions that we can think of with this? Perfect. Getting a lot of positive feedback. So I love it. Tony the Tiger is making an appearance. He feels great about this stuff. Good. All right. Well, let's keep trucking then. When we're using our microscopes, there are 2 things that we can do with the microscopes. The first thing that we are always doing with microscopes, because we just talked about this, is magnification. So magnification, if I were to define that, it just means making something bigger. With our microscopes, the goal is to make things bigger, absolutely. But we don't just want to make them bigger, we also want to make sure that they're clear. So this idea of resolution is basically how clear something is. So I've got 2 pictures down here. Let's say we started with this letter E that I just drew here in the middle. Both of these pictures next to it show something that's been magnified, something that's bigger. Bigger is great, but if I don't have resolution, if I'm not clear, it doesn't help me. So my picture over here on the right shows me magnification without resolution. My picture over here on the left shows me magnification and resolution. We want things to be big and we want them to be clear. That's the way that microscopes work. When we're looking at slides, there's an important idea for us to consider called depth of field. So every slide that you put on your microscope has a 3D amount of tissue inside of it. And a good way to think about things as 3D, we have a slide in our lab that literally has 3 little threads stacked on top of it. So there's a 3D space inside your slide where the tissue is found. The amount of that 3D space that you can focus on at one point of time, that's something called the depth of field. So imagine that your slide is a swimming pool. How deep can I focus without things getting blurry? That's the depth of field. When you have the scanning objective in place, the red objective, your depth of field is really big. So if we were looking at this slide in the lab, all three of these fibers, we would be able to see them in focus. We have a big depth of field. When I put that yellow objective into place, the low power objective, my depth of field goes from this deep in the swimming pool to this deep in the swimming pool. Maybe now I can only focus on two of those three fibers because the other one is too deep. I can't keep it in focus anymore. And if I put that high power objective in place, the blue in place, I may only be able to focus on one of those threads or part of one of those threads. So the idea of depth of field is as you go from a low power objective to a higher power objective, the more magnification that we do, the less amount of space is going to be in focus. So when you're looking at the slides in labs, sometimes we'll have to play with that fine adjustment knob to change our depth of field so that we can see what those cells look like, the depth of field. Another idea with those slides is something called the field of view. So not only are your slides 3D going down like a swimming pool, they're also a big wide space like a field. So when we talk about the field of view on a microscope slide, what we're talking about is how much of that slide can we see when we're looking through the ocular lenses. So imagine that I've got a slide, it's a boring slide, it just has this little black dot on it. When I'm looking at it with the scanning objective in place, I can see all of this space around it and I can see the dot in the middle. When I put my low power objective in place, notice that the spot itself got bigger, it got zoomed in, and the amount of space next to it that I can see is smaller. My field of view got smaller. I'm narrowing in. And if I go all the way up to my high power objective, I can't hardly see anything that's outside the spot. The field of view, how much of that slide you can see at once. As magnification goes up, the amount of space you can see on that slide, your field of view for that slide, gets smaller and smaller. One of the biggest reasons this is important is when I put a slide on my microscope and I get it focused on the red objective, I want to make sure that what I'm looking for, in this case, this black dot here in the middle, I want to make sure that that's in the middle of my field of view. Because if it's not, if this is what's in the middle of my field of view, I'm going to zoom in onto that X. Whatever is in the very middle of your slide, when you go from one objective to the next, you're going to zoom right into the middle. So make sure that if you want to see this black dot, make sure that black dot is what's in the middle of your field of view. If you're more interested in the gray stuff, it doesn't matter. Go over somewhere in the gray stuff. But when we start working with those slides next week, whatever you're looking for, that kind of tissue, that structure, make sure that's in the center of your field of view. Because all you're going to see when you go up in magnification is that stuff in the very center. So these are the terms that we just talked about a little bit before. Remember that the depth of field, this word depth, tells me how much of the 3D space can be in focus at once. The field of view, so how much you can see of the slide looking from the top. And then we talked about, remember how our microscopes are parfocal. So we stay mostly in focus. If we do a good job focusing once, we're going to stay mostly in focus when we go from one objective to the next. And then, of course, that working distance. The working distance was the amount of space I had to make sure I don't crunch my slide, the working distance. I want to briefly show you, since we're not in our lab class together, let me pull up. Oops. There is a virtual microscope available. I'm going to copy the link into the chat, so if you'd like to track along with me. This virtual microscope, loading very slowly. Okay. Ooh, there we go. This virtual microscope is going to allow us to explore a couple of these ideas that we talked about. So the link should be coming through to you now if you'd like to play with the virtual microscope on your computer. I'm also going to show you here on mine. So I'm going to click on this website. I'm going to click on the Explore area, and we're going to explore the microscope together. So we're looking at a compound microscope that looks very similar to the ones in our lab. Remember that we have those ocular lenses at the top that we look through. You can see the objective lenses that are here hanging down. Here's that mechanical stage where we put the slides onto our adjustment knobs. So we're going to play around with a couple of different slides to explore how the microscope works. So first, we're going to look at a random sample slide with the letter E on it. So it's not a human cell, it's not anything else. We just put this letter E slide onto our microscope. When I first put a slide on my microscope, I'm always starting with the red objective, the scanning objective, which is the one that made things 4 times bigger. So I am magnifying things with my red objective. Because my red objective is in place, I can or cannot use the coarse adjustment knob. I can or cannot, since my scanning objective is in place. What do we think? Exactly. I can. And in fact, I want to. When I first put a slide onto my microscope, I want to use that coarse adjustment knob. So I'm going to use that coarse adjustment knob. I'm going to get it almost all the way there. Hey, that looks pretty good to me. If I have some things I need to adjust, I can use that fine adjustment knob to make it extra crisp. Perfect. I've got it focused with my scanning objective in place. Now I'm ready to move up to my low power objective. So now I'm going to rotate in yellow into place. Notice that I zoomed right in to the middle of what I was looking at, the middle of my field of view. And notice how this is mostly in focus already. Now that I have my low power objective in place, now that yellow is in place, I can or cannot keep using the coarse adjustment knob. I've got that low power objective in place now. Can I keep using this one? Exactly. I cannot, because if I do, it's going to mess things up royally. Remember, it should be mostly in focus already when you go from the red to the yellow. So we shouldn't need to use the coarse adjustment knob because it should mostly be focused. But even if it's not mostly focused, it's too big of a risk for me to break that slide. So if I need to fix anything, I'll use that fine focus, made it a little bit more crisp with that fine adjustment knob. Now I'm going to go all the way up to the high power objective, all the way up to blue, and see how I can only see this little section of the letter E here. I can't tell that it's a letter E anymore. At this point, you better not touch that coarse adjustment knob. You'll completely lose it. You won't be able to find it. If I need to, I can use this fine adjustment knob, or in this case, what might help me is adjusting that light a little bit. So we can use that light adjustment knob that's on the sides on ours, or you can use that iris diaphragm under the stage. What I will tell you in class next week when we start using the slides, if you get your slide up to the yellow or the blue objective, you say, I can't get it in focus. I'll tell you, okay, let's go back to yellow and let's make sure it's in focus on yellow. Because if it's not in focus on yellow, it's not going to be in focus on blue. If we can't get it in focus on yellow, I'm going to say, hey, let's go all the way back to red. There's no harm in starting over, because if I have my coarse adjustment way out of focus on red, when I go up to yellow, I'm going to have a really hard time getting this in focus because it's all messed up way at the beginning. So always make sure the most important thing you can do with a microscope slide is to make sure that you get it focused on our scanning objective. Make sure it's working on red. That's going to allow us to get it in focus on other levels as well. I know a virtual microscope is different than an in-person microscope, but how do we feel about potentially using microscopes? You tracking with me? Does this generally make sense? Perfect. And like I said, we're going to do some practice next week. We've got, I think, 5 different slides we're going to look at next week. So we'll get to do all kinds of microscopy together. But again, if you want to play around with slides, that link that I put in there for you for the virtual microscope would be a way that you could do that. All right, so let's assume we are at the end of lab and it's time for us to put away our microscopes. Let's talk about how we would do that. So you've got a bunch of true false statements in your lab packet. I'm going to help you with completing these statements. The first thing we do when we finish looking at our slides is we're going to drop our stage as low as possible. So we want the working distance, the space between the objective and the stage, to be as big as possible so we don't break any slides. So the stage is dropped, not raised, it's dropped as low as possible. We're going to pull our slide off of the stage. We don't want to leave slides left on our microscope. So we'll take that slide off of the stage. We'll undo that stage clip to get it out. We're going to make sure that the scanning objective, I'm just going to write red here, that the scanning objective is what's pointing down. If the high power objective was what was pointing down, that's the really long one. If we leave that pointing down, better chance we're going to break our slide. So make sure that the red objective, the tiny one, is what's pointing down. Before you unplug that microscope, we definitely want to turn it off. So turn it off, then unplug it. On the back side of the microscope, we have a space in the arm for you to tuck that power cord into. So we'd be tucking it into there. And we're definitely using 2 hands to transport the microscopes. 1 hand under the base, 1 hand on the arm. We'll get really good at moving microscopes. We're going to be using microscopes almost every week this semester, starting next week. So lots of microscope work coming at you soon. We'll get really good at storing them, and we'll get really good at figuring out who doesn't know how to store microscopes, because what's going on with this one? The stage is all the way up, the scanning objective isn't in place. So we'll get really good at calling other people on their storage woes too, right? It's only good if you can call other people on it too. All right. Last thing that I'm going to chat with you about before we do our group work is the organelles that we find inside our cells. So all of our cells in the body have a variety of different organelles. We have talked about these in lecture on Monday. So let's go through and work on identifying them. You will have to know for lab both how to identify them and what they do. So make sure we're familiar with both. Let's start with the big organelle that we find in the middle of our cells here. This big organelle in the middle of our cells that stores our genetic information. What is this organelle here in the middle called? What would we say this is called? Do you remember from lecture? Exactly. We were frantically flipping through our notes. So this big central organelle in our cell is called the nucleus. The nucleus. Inside of the nucleus, there's something we didn't talk about in lecture, but we do need to know it for lab. So this little marble you see in the middle here, this little white spot that we see in the middle here, this is something called the nucleolus. Nucleolus. Sorry, that looks so terrible. The nucleolus is like a nucleus inside the nucleus, the nucleolus. While the nucleus stores the DNA for the cell, the nucleolus is what the cell uses to make ribosomes. So ribosomes are organelles that I can see each of these little white spots that you're seeing on our model here. Each of those white spots are ribosomes. In this model over here, we've got an awesome little conga line going of those ribosomes. Ribosomes are found in two places. They're either found, like we see in our conga line over here, they're found free floating inside the cytosol, inside the soup of the cell, or they're found attached to that thing we talked about in lecture called the rough endoplasmic reticulum. Rough ER. The rough ER is all of this stuff that's blue and has the white spots on the outside of it. So the rough ER are those membranes that have ribosomes on the outside of them. Those ribosomes build proteins, whether they're by themselves or whether they're on the rough endoplasmic reticulum. So the two organelles that make proteins in your cells are the ribosomes by themselves, and then I'll highlight on this model as well, these little spots that are on the outside of my purple membranes. That's my rough ER over here. When we're talking about the smooth ER looks just like the rough ER, except it doesn't have ribosomes on it. Full disclosure, I don't think that the cell on the left really has good smooth ER. Some people will say that this is supposed to represent the smooth ER down here because it has no ribosomes on it. If I'm going to ask you about the smooth endoplasmic reticulum, full disclosure, I'm going to use this right here. We can clearly tell that this is the endoplasmic reticulum, and we can clearly tell that it does not have red spots. So the smooth endoplasmic reticulum, I can see it there. I can see it over here, these flat sacs. I can see more of them down here. If you see these flat sacs without the red dots, that's the smooth endoplasmic reticulum. The sacs that have the red spots, that's the ones that are the rough ER. Couple other organelles that are the same color on both of my models. We've got these ones here that are orange. I can see them here and I can see them over here. These organelles are, as we talked about in lecture, the powerhouse of the cell. What would be the name of these orange ones that we see? Our cell's powerhouse, as we all learned it. Exactly. I was mean to make you type that, right? You totally could have just typed mito. I would have knew what you meant. The mitochondria, absolutely. The mitochondria are the powerhouses of the cell. So the mitochondria seen in yellow on both of these models. The other thing that's the same color on both of my models are the pink ones that I see. The pink ones are called the Golgi apparatus. So here's that Golgi apparatus. I think I mentioned it in lecture, but the Golgi apparatus is a lot like a post office, if you will. We send proteins there, it packages them up, and it ships them to where they need to go inside your cell or outside your cell. So the Golgi apparatus, packaging, modifying proteins, and sending them places in the cell. The only other organelle that we want you to be able to identify on our models are these churros that I see right here, or these little licorice pieces. That's the same one that I see over here. These are things called the centrioles. The centrioles. And the centrioles will be very important next week because this is the organelle that helps you to divide your genetic information when your cell is dividing. We need the little churros or we need the licorice to help us to pull apart our DNA when we're dividing. There are a couple of other organelles that you don't need to identify, but you do need to know what they do. I think we talked about these a little bit in lecture as well. So if I were to guess, the yellow bubbles, I would say, are lysosomes. That's what I would guess. But I have nothing that tells me that for sure, so I'm not going to ask you to identify it. When we talk about lysosomes, remember that those break down waste. They do digestion. That's the lysosomes. If I were to guess, I would say that these black dots are the peroxisomes. Remember that peroxisomes deal with free radicals in your cells. So I kind of like to equate them to hazmat. They come in and they clean up really bad messes. They keep you safe from those free radicals. So lysosomes and peroxisomes, you only need to know what they do. You do not need to identify them on the models because we don't have something that tells us for sure which is which. With that, I am done covering the content I'm going to cover with you. You have resources in the guided lesson to help you with identifying which organelles do which task. So I'm not going to take our lab time to cover this with you. But I do want to give you a chance to ask any last minute questions before we dive into our group work. So what questions do we have about any of the content that we covered during today's class? Any last minute thoughts before those groups? We are very quiet. That's okay. What is my favorite organelle? Ah, that's a good question. Hmm. I need to think about that more thoroughly. I'm tempted to say the mitochondria just because, it's really good to have energy. Um, I also kind of like the way that it has, so the mitochondria looks funky. Let me pull up my pointer here. It looks funky because it actually has two layers of membranes inside of it. So that's kind of cool to me. So if I were to pick it right now, I'd say the mitochondria. That's a good one for sure. Also, though, I have to give some credit to your ribosomes. We're going to talk a whole lot about ribosomes in unit number 2, making those proteins for you. So 2 heavy hitter organelles, as they say. All right, let me shut off our recording and we'll talk about the group work.